comes the sun.
Welcome to Miss Whitelaw's class. We're working on a jigsaw experiment. And here we have a lost specimen in biology, or life in general.
Miss Whitelaw, I don't see anything. I see the dark of night. I already adjusted everything. Ugh, I'm gonna die. Help, please.
Alexis, you forgot the two most important steps. First of all, this isn't even connected.
Oh, come on.
Second of all, you need to turn this on.
I don't see anything.
Alexis, can you see anything now?
Yeah, I don't know. Well, oh yeah, look, I can see that. Look.
Keep moving the fine adjustment.
Ooh, I can see something.
Look, Alexis, interphase is the first phase that cells go through in order to divide.
This stage is divided into 3 substages. First one being G1, which is where the cell doubles in size and so do most of the organelles. The second one being S, which stands for synthesis, and it is where the DNA replicates. And G2, the last one, is where rapid growth occurs, but basically it is just a double check before it goes into prophase. Interphase is very important because the cell spends 90% of its life in this stage.
And as you can see, the organelles present here are the nuclear membrane, the centrioles, chromatin, and the nucleolus.
But Miss Whitelaw, how do cells divide? I don't get it.
Well, so there isn't any DNA overload, and also to maintain a favorable surface area to volume ratio so they can function efficiently. And that way, the cell membrane will be able to keep up with the organelles and their needs.
Okay. The next phase is prophase.
Music floats throughout the land.
And here, the nuclear membrane starts to break down into vesicles.
Doesn't each chromosome duplicate and consist of 2 sister chromatids?
Of course, Sophia, and they're also held together by those little blue buttons called the centromeres. And also, the centrioles move to opposite poles.
And after prophase comes metaphase. And look, now their nuclear membrane has completely vanished.
That's the way to go. It's looking like a good day. Won't you come along?
And here, the chromosomes align at the equator and are held in place by microtubules attached to the mitotic spindle, also known as spindle fibers, into a part of the centromere like tiny Spider-Mans.
What about Batman?
Alexis, he's not in this story. The next phase is anaphase, and this is when the spindle fibers pull the chromatids apart and move to the opposite sides of the cell, and the centromeres also divide, and the chromosomes become sister chromatids.
How we gonna reach down? It's easy if you try.
Sophia, do you know the next phase?
Yeah, the last step of mitosis is telophase.
If you give a little love.
This is when the spindle fibers disappear and the cell membrane starts to pinch in on itself, creating a cleavage furrow.
Cleavage.
Then, the nuclear membrane starts to form around each of the chromosomes, which return to the form of chromatin, and the nucleus reappears.
But Miss Whitelaw, what prevents a cell from growing?
That would be when cells come in contact with other cells. Sophia, would you like to explain the last step of cell division?
Sure, it's cytokinesis. This is when cytoplasm splits completely, forming 2 new daughter cells.
And what happened to mommy cell?
Well, from the parent cell came these 2 daughter cells.
panic. Drink this delicious protein-filled cyclinate. Your cells will regulate and function fantastically. Whoops! Sorry! Buy now to Mercados. If you can't find it there, call 855-661-123.
Why do you need this product? If your cell cycle is not regulated, your cells will start to clump and create a tumor, which could potentially result in cancer, which could be malignant or benign. So don't take chances, and we suggest that you drink cyclinate.
Alexis, did you learn anything? Yes, guys, thank you so much. I love Miss Whitelaw's class. Yay!
Okay, Alexis, that's so much.